Dear friends, today we are going to talk about the preservation potential of organisms and morphology of Gastropoda. The word fossil has no exact definition, but it is loosely taken to mean any organism whose remains or traces of organisms or remains are preserved in some kind of sediment. The term is derived from the Latin fossare, that means to dig. The definition implies nothing about the age. Fossils range from those that are about 3 billion years old to those that are preserved in lime saturated water deposits only a few days ago or even hours ago. The study of fossils is termed as paleontology, which is a word derived from the Greek that literally means knowledge of ancient things. However, for more than 2 centuries, the word paleontology has been restricted to the study of formerly living, that is organic, not inorganic remains or traces. The word paleobiology is sometimes used as an alternative. This article is designed simply so that you may find your own way through the ensuing contributions. Very few fossils are found that show no changes from the original organisms. There is a very approximate correlation in many cases between the age of the fossil and the degree of change, but age is in itself no guide. For example, a fossil may be distorted by tectonic activity within a few 100,000 years of its deposition, whilst other rocks and their contained fossils undergo little tectonic changes over long periods. For example, the Lower Cambrian shale in the St. Petersburg area of Russia, which is some 430 million years old, is so soft that it can be dug with a spade, and the marine shells preserved within it can be washed out and look not dissimilar in preservation from those found on a beach. The term diagenesis describes the changes within both rocks and fossils as the sediments become dewatered and chemical transformations occur. Voids from microscopic size upward, present both in the original organism and in the surrounding sediment, are usually filled during diagenesis as a result of mineral charged fluids circulating through the rock. This may occur at any time from soon after the fossil was deposited to many millions of years later, when the entombing rock is subjected to sedimentary pressure or tectonic events. Replacement of the chemicals in the fossils themselves is widespread. For example, the calcium carbonate or CaCO3 of a brachiopod shell is often replaced by silica, that is SiO2, during diagenesis. Fossils may also be hugely distorted from the original shape of the organisms by tectonic processes. In addition, the fossils may dissolve and their chemical contents may form other substances. For example, all fossil fuels such as crude oil and natural gas are made up of the concentrated remains of fossil plankton, and coal is formed from the now usually unrecognizable remains of fossil plants. Another process is termed as disarticulation. Obviously, if an animal or plant has only a single hard part, such as the shell of a snail, then that may become weathered or broken, but there is still only the one shell. However, if an organism has more than one hard part, ranging from the 2 shells of a bivalve mollusk or brachiopod to the several hundred bones found in a mammal skeleton, then the process of the transport from its place of death to its final burial place, where it will eventually form a fossil, will often cause the organism to break up or disarticulate. The degree of disarticulation often depends on how strong or weak the original articulation was. For example, some brachiopods have weak hinge structures, and others, such as the common Silurian genus Atripa, have complex interlocking mechanisms between the two valves and are thus very commonly found fossilized with the two valves still close together. Coming to the biostratonomy, the term biostratonomy was proposed by Wiegelt 1919 and was originally defined as the study of the manner in which the fossils become oriented and arranged in the rocks. Today, it has a broader meaning but still embraces the basic concepts developed by the Wiegelt. The study of biostratonomy begins with the death struggle of an organism and ends with the final burial and arrangement of dead or dying animal or its disarticulated remains. The fate of materials produced and discarded during the life cycle of organisms must also be included in biostratonomy. The chance of an organism being preserved as a fossil is dependent upon the conditions of sedimentation, which can in turn be related to the paleogeography of an area. Animals living in high altitudes which are subject to active erosion are seldom preserved. On the other hand, those that live in low-lying areas or in streams, rivers, swamps, seas or oceans stand the best chance of being covered by sediment and eventually being preserved. The first systematic observations were made by Walther, followed by Wiegelt. In general, conditions of preservation for aquatic animals are better than those for land dwellers. A similar relationship applies to sessile, that is attached or infaunal animals when compared to vagile animals. In principle, every organism is preservable no matter what the proportion of its hard and soft parts. Preservation in the final analysis is dependent only upon the dominant physico-chemical and biological conditions to which an organism is selected. In most cases, the fate of both hard and soft parts is different, and as a rule, the soft parts decompose and decay, leading to a relatively large loss of body material. This process commonly leads to a more or less complete segregation of the more resistant hard parts, which can be broken or macerated, depending upon the chemical or physical processes to which they are exposed. Formation of steinkerns and early diagenesis. The body of an organism can be completely preserved as a body fossil if it is destroyed within the sediment and if the sediment is sufficiently lithified and no secondary minerals are being precipitated. A void is an impression of the outside of the destroyed object. Solution of, for example, bivalve shells and subsequent filling of such voids by druzy materials such as calcite, dolomite or siderite is relatively rare. If the sediment is not sufficiently competent to maintain these voids, they may collapse. However, if the shells initially possesses an internal void or if such voids are created during early diagenesis by removal or decomposition of soft parts, they are commonly filled by sediment. It is by this process that true steinkerns or molds are developed, reflecting the internal morphology of the shell. They are produced by many types of the sedimentary infilling and have an amazing variety of shapes and textures. Frequently, they undergo internal sedimentation and develop geopetal structures. Commonly, small fossils and burrows are found in them. A study of steinkerns allow important conclusions to be drawn with regard to the life habits of an individual organism and the particular conditions of sedimentation. For example, calcilutite and pelletier limestones can occur next to many limestones or marly limestones and shell debris deposits. Multiple phases of fine-grained sedimentation can occur within shells by recurrent water movement. Material in suspension then settles within the shells, especially if the currents flowing out of the shells are weak. In planispiral ammonite shells that are being filled with sediment through their apertures, the sediment in the camerae becomes increasingly finer and more homogeneous towards the apical end. Many camerae receive no sediment and remain empty, and most shells are not entirely filled. A hole at the apical end of the shell would have to exist. When the shell substance is gradually destroyed, the surrounding sediment may settle slightly and the steinkern or the mold may be pressed against the impression made by the outer surface of the shell, so that this is then imprinted on the steinkern. Such structures are called as the pragiekern or sculpture steinkern. Their analysis helps in the interpretation of early diagenetic process in the surrounding sediment as well as the manner of dissolution and decomposition of the organism itself. Sculpture steinkerns are most frequently formed as the result of the destruction of the organisms with calcareous hard parts, in particular bivalves. The dissolution of the shell material can begin on either the interior or the exterior surface of the shell. Dissolution has been shown to be extremely strong in fractured shell material. The synchronous dissolution of the shell material can also lead to the decrease in the shell thickness and the loss of sculpture. Shells are most easily reduced in the thickness at the ambonal region where they were originally thickest. This type of shell destruction is mostly the result of process occurring outside the sediment prior to burial. In both these cases of shell destruction or reduction, it is thought that the conchiolin and the periostracum are destroyed. In oxygen-rich environments, the periostracum is generally destroyed first. The inorganic constituents remain. In oxygen-deficient environments, especially those with high concentrations of hydrogen sulfide, destruction begins with the dissolution of calcareous components. The oxidation of sulfides releases sulfuric acid, which dissolves calcareous material and hinders additional bacterial destruction of organic substances, as decay-producing bacteria are extremely sensitive to the presence of oxygen. In anaerobic environments, the periostracum protects the underlying shell material, but there is a considerable difference in the solubility of the prismatic and the nacreous layers. The prismatic layer of the shell secreted by the mantle edge is generally calcite, whereas the nacreous or porcelaneous layer consists of more soluble aragonite, which is secreted by the mantle surface. The shells of the snails and Dimarian bivalves, whose shells are composed entirely or predominantly of aragonite, are therefore most commonly preserved as steinkerns or molds. The shells of monomyrians are predominantly of calcite and their shells are generally preserved entirely. If the voids created by solutions of skeletal material are preserved, the solution must have occurred after lithification of the sediment. Occurrences of the sculpture steinkerns show that the solutions of the shells proceeded outwards from the inner side. They can only be formed if the sediment has not become entirely lithified. When the void can be closed under the weight of the settling sediment, during this process, the steinkern moves outwards with the progressive solution of the inner side of the shell. Imprinting of the outer ornamentation on the steinkern occurs only after the shell substance has been completely removed. Aragonite can be replaced in a skeletal material by the action of the marine boring algae and probably fungi and bacteria. Algal filaments penetrate the shell material centripetally and eventually die and decompose also. The resulting cavities can be filled with micrite cements. When this process is repeated, as have been observed in mollusk shells from the Bahamas, a micrite envelope or rind will develop around the shell, which for unknown reasons is not dissolved during the diagenesis. Thus, an impression of the original shell is preserved, which latter may be filled with calcite crystals. Fossils can also be preserved in lava flows. For instance, tree trunks which were surrounded by lava can leave many details of the bark clearly imprinted in the surrounding rocks, and in some cases the cell structure of trees has been preserved. Even a rhinoceros has been preserved in the basalt. Chappell et al. 1951 can demonstrate it much better. Another example of particular interest is the metallofact, which is a myriapod rapidly covered by molten aluminium, thereby preserving its shape. Gastropods, after insects, are the most highly diverse animals on the Earth's surface. Well-known examples of the gastropoda are the snail, the whelk and the cowrie. The bilateral symmetry so characteristic of the lamellibranchs is generally to a large extent obliterated, owing to the twisting of the visceral mass and the atrophy of some of the organs on one side of the body. There is a distinct head which bears one or two pairs of tentacles and usually two eyes also. On the ventral surface of the foot body is the foot. This is usually large and so-like and used for crawling, but in the heteropods it is in the form of a flattened fin, and in the pteropods it is wing-like. The mantle is never divided into two lobes. Respiration takes place in some cases through the skin, but generally by means of lung cavity or by gills. The latter are placed in a sac formed by the mantle. Sometimes they are present on both sides of the body, but usually the original left gill has disappeared. In some forms, the mantle at the opening of the gill sac is produced into a tube known as the siphon, by means of which the water passes into the gills. The heart is on the dorsal surface and consists of a ventricle and usually one, but in some cases two auricles. In many forms, the gills are placed in front of the heart, but in others behind it. The mouth is at the anterior end of the body and the anus is near the opening of the gill chamber. On the floor of the cavity of the mouth is a dental apparatus known as the odontophore. This consists of a cartilaginous and muscular ridge on which rests a chitinous ribbon, that is the radula. The radula bears numerous teeth placed in rows and serves as a rasping organ or an organ by which the food is chewed. The arrangement of the teeth varies in different genera and is of considerable importance in classification, but since the radula has never been definitely recognized in the fossil forms, it can only be used by the paleontologists in case of the genera which have existing representatives. In most gastropoda, except the carnivorous genera and the heteropods, there is also one or two horny jaws in the upper part of the mouth which are used for biting. The nervous system consists of ganglia which are connected by nerve cords. Typically, there are three pairs of principal ganglia: the cerebral placed above the esophagus and the pleural and pedal placed below it. A visceral nerve cord which may bear ganglia comes off from the pleural ganglia and forms a loop ventral to the intestine. In some gastropods, this loop is simple, but in others, that is the prosobranchiata, one side is bent over so that the loop forms a figure of 8. In some gastropods, the sexes are separate, but others are hermaphrodite also. In the majority of the gastropods, a shell is secreted by the mantle. In a few forms, as for instance the slugs, it is internal. but usually it is external and covers the visceral mass. The shell consists of a single piece, hence said to have univalve. In the limpet, that is the patella, it has the form of a hollow cone, but in most cases it consists of a long tube open at one end and tapering to a point at the other. This tube is coiled into a spiral, generally screw-like, each coil being termed as a vole. In a few genera, for example Vermetus siliquaria, the voles are separated, but as a rule they are in contact, the line between the two contiguous voles being known as the suture. All the voles except the last together form the spire of the shell, the point of which is termed as the apex. The last vole is nearly always larger, frequently much larger than the one preceding, and the part of it farthest from the apex is called as the base of the shell. The spire varies in the form or in different genera and species. Sometimes it is composed of a larger number of voles, sometimes of few, and it may be long, short or depressed. Occasionally all the voles are in one plane. The angle of the spire, that is the spire angle, consequently varies. This is measured by lines drawn from the apex to the base of the shell on opposite sides of the exterior of the voles. The coiling of the shell is usually dextral, so that when the apex of the shell is pointed away from the observer, the aperture will be on the right hand side. In a few cases it is sinistral, for example Physa, when the aperture will be on this left, or sinistral forms may occur as the spots in the dextral species, or they may be characteristics of a species or occasionally of a genus. Frequently the inner parts of the voles coalesce and form an axial pillar extending from the apex to the base of the shell and known as the columella. In other cases the inner parts do not fuse, and in the place of the columella there is left a tube-like space extending from the base of the shell a greater or less distance towards the apex. This space, which opens at the base of the shell, is called as the umbilicus. When there is a columella, the shell is said to be imperforate. When instead there is an umbilicus, it is perforate. The opening of the umbilicus sometimes becomes partly or completely filled up with callus. The animal is attached to the columella by means of a muscle, the contraction of which enables it to withdraw completely into the shell, but when not retracted, the coiled visceral mass only is covered by the shell. Usually the cavity of the gastropod shell is continuous from the apex to the aperture, but in a few cases partitions are thrown across the earlier parts of the shell, forming chambers which remain empty. The form of the aperture varies considerably in different genera and is of great importance in classification. In shape it may be circular, oval, elongate, oblong, etc. Its margin is termed as the peristome. The outer part forms the outer lip, the inner part that next the columella, the inner lip. Coming to the fourth module, as the gastropod crawls along, the shell is carried on the dorsal surface of its body with apex directed backward and upward, and the aperture downward. Consequently, the part of the aperture farthest from the apex is anterior, the opposite near the apex is posterior. Sometimes, as in Natica, there is no break in the peristome and is then said to be entire or holostomatous. In other cases, the anterior border is notched or produced into a tube in which the incurrent siphon is placed, and these forms are said to be siphonostomatous. Sometimes there is also at the posterior border another notch or a canal in which the excurrent or anal siphon is placed. The two lip may be thin and sharp or thickened. Sometimes it is curved outwards and is then said to be reflected, or it is curved inwards inflected. Its margins may be even or granulated or produced into processes or grow outwards to form a wing-like projection. In some genera, a shelly deposit termed as the callus is secreted by the mantle on the inner lip and adjoining part of the last whorl. Many genera have a calcareous or horny plate known as the operculum attached to the dorsal part of the posterior end of the foot. This is so arranged that when the animal withdraws into its shell, the operculum more or less completely closes the aperture. It probably represents the byssus of the lamellibranch. The operculum is seldom preserved as fossils. Its form varies considerably in different genera. In some, for example turbo, it is of very large size with the inner surface flattened and the outer convex. It may have a spiral structure and is then sometimes formed of a large number of whorls, that is multi-spiral, as in Trochus, or of a few whorls, that is the plosispiral, as in Littorina. When not spiral, it may be concentric if growth takes place equally all around. It is then marked with the concentric lines, the nucleus being clearly or nearly central, as in viviparous, or it may be unguiculate or claw-shaped when the nucleus is at the apex, as in the Fusinus. The form of the shell in the spiral gastropods varies considerably depending upon the arrangement of the whorls in one plane or in a helicoid spiral or the spiral angle, on the number and shape of the whorls, on the size of the last whorl, and whether it conceals the earlier whorls or not. The chief types are the following. Number 1, discoidal. All the whorls are nearly or quite in one plane and all the visible on the exterior, as in planorbis. Number 2 is conical or trochiform, conical with a moderately acute spire and flat base. The whorls increase in diameter uniformly and have nearly flat surfaces, for example Trochus. Number 3 is turbinate, that is conical with a convex base, as in turbo. 4th is serrated, a spire is long, very acute, formed of numerous whorls which increase in diameter slowly and uniformly, as in Turritella. 5th is fusiform, spindle shaped, thickest in the middle and a tapering to each end. The spire is elongated and the base of the last whorl is produced into a long neck, as in Fusinus. Coming to the 6th one, that is cylindrical. After the first few whorls, their diameter remains constant or may decrease near the anterior end of the shell or papilia. 7th is globular. The spire is short in this case, last whorl large and rounded, as in Natica. And 8th is convolute, when the last whorl is very large and convex and covers all or nearly all the others arid. The aperture is consequently as long as the shell, as in SWAYAMPRABHA. The surface of the shell is frequently ornamented with spines, knobs, ribs or striae. These are said to be spiral when they run parallel with the sutures and transverses when they cross the whorls from the suture to suture. In some genera, for example Murex, rows of spines or lamellar processes extend across all the whorls from the apex to the base of the shell, forming what are termed as varices. The surface of the shell in recent gastropods is generally colored, often variegated. In fossil examples, the color has nearly always disappeared. But in few specimens from various formations, even as early as the Carboniferous, have been found showing the color more or less perfectly preserved. The shells consist of an outer chitinous layer and of a calcareous layer, usually aragonite, which is thick and porcelaneous.